Okay, good evening, everybody. My name is Alan Armstrong, and I'm really pleased to welcome you all here to today's inaugural lecture of my colleague, Professor Matt Fuchter. Inaugural lectures provide official recognition and celebration of a promotion to professor. So it's really pleasing that so many of you are able to join us for this great occasion, including many members of Matt's family, who it's wonderful to see here today as well. Now, before I introduce Matt, I've been asked to make a quick housekeeping announcement. I don't think Matt is planning any fireworks or anything like that in his lecture, but if the fire alarm does go off, please leave the theatre via one of the marked exits, and then the assembly point is out on Exhibition Road. But as I say, hopefully that won't be needed. But anyway, back to Matt. Many of you know him very well, so he doesn't need much introduction, but I'll give one anyway. A bit about his background. So Matt obtained his first degree as a first-class honours in chemistry from the University of Bristol in 2002. And then he came here to Imperial, where he obtained a PhD degree in 2006 under the supervision of Professor Tony Barrett. And then he had a brief spell as a postdoc in Australia at the Commonwealth Scientific and Industrial Research Organisation, CSIRO, where he worked with Professor Andrew Holmes. And I'm really delighted that Andy Holmes is here today and will be giving the vote of thanks after Matt's lecture. Andy, amongst many other distinctions, is the immediate past president of the Australian Academy of Science. So welcome, Andy. After his postdoc stay in Andy's lab, Matt came back to the UK, back to London, in fact, for a brief stint at the School of Pharmacy in an independent position. But he didn't stay there long because we quickly snapped him up to come back to Imperial a year later to a joint appointment between the departments of chemistry and medicine. And since then, his career has advanced really rapidly. So he was promoted to senior lecturer in 2012, then reader in 2015, and now full professor from 2018. And as a fellow organic chemist and colleague, it's been really wonderful to see Matt's career flourish because he's achieved an amazing combination of really important and interesting fundamental science, but he's also led the way in showing how the ability to make molecules and predict their properties can lead to some really important applications, ranging from materials to medicine. And a great example of that has been his work as part of a team between chemistry and medicine that's developed a new cancer drug that's currently in clinical trials. And that work was recognized with the Imperial College President's Medal for Excellence in Innovation and Entrepreneurship back in 2017. And he's also won several other prestigious international prizes, like the Royal Society of Chemistry's Harrison-Meldola Medal in 2014, and the Tetrahedron Young Investigator Prize for Bioorganic and Medicinal Chemistry in 2018. And he's done all of this whilst playing his part for the community. He's currently an elected member of the Royal Society of Chemistry's Organic Division Council, and also the UK representative to the European Chemical Society's Division of Organic Chemistry. So, amazing set of achievements. Congratulations, Matt. And congratulations on your professorship. And without any further ado, I'm delighted to invite you to give your inaugural lecture entitled The Central Science Using Molecules to Develop Materials and Medicines. Congratulations, Matt.
Well, thanks a lot, Alan, for the very kind introduction. I have to say this is a wonderful, a humbling, and a slightly daunting opportunity I have tonight to give a lecture where I have my friends and family, past and present members of my research group, collaborators, colleagues, staff, student, maybe even some members of the public. So what I've tried to do in my talk today is make sure it's relatively accessible. There will be some molecules in my talk. This is a chemistry talk, after all, but hopefully there will be something that everyone can take away tonight. Now, as you can see from the title of my talk, I borrowed a phrase that's often used to describe chemistry, the central science. This is a phrase that was perhaps best popularized by a textbook of chemistry, But there's been a number of studies over the years that have sought to position the sciences relative to one another and have placed chemistry in a central position. So here's a study over here from the 19th century where you can see that chemistry bridges a gap between the physical sciences and the life sciences. Here's a much later study where they claim a central position for chemistry because of the number of branch points it has with the other scientific disciplines. Now, personally, I like the description of chemistry as a central science, not because I'm trying to claim that chemistry is more important than the other sciences, but instead, for me, being in a central position really nicely explains how chemistry and how chemists can interact and collaborate so cleanly with a wide variety of other disciplines. And through that collaboration, generate a number of different multidisciplinary research topics. And indeed, it's that kind of multidisciplinary research that underpins the research I do here at Imperial College. Now, when we as scientists are asked to justify our motivation for doing certain scientific areas of study, we're often asked to place ourselves in one of two camps. We're either a basic scientist, so we're interested in furthering scientific knowledge without necessarily being concerned in how that knowledge may be used. Or alternatively, we're an applied scientist, so we use already existing scientific knowledge for a specific application. Now, personally, I've never particularly liked this framework and the extreme nature of these two definitions. I've never thought that it fits the type of science that I'm most interested in. So what is it that I do? Well, first and foremost, I'm an organic chemist. So that means I'm interested in molecules that are predominantly made from the element carbon. Moreover, I'm a synthetic organic chemist, which means I develop methods and can use existing methods to construct new chemical bonds and so build new molecules from simplified starting materials. Now, armed with that skill set, you might ask, well, why do we make molecules? What can we learn from that process? Well, one of the long-standing reasons we make molecules is to better understand molecular structure, the dynamics of that structure, and how molecular structure relates to molecular properties. One of my scientific grandparents was Sir Derek Barton. He spent much of his career here at Imperial College and won the Nobel Prize for his contributions towards understanding molecular structure. Alternatively, we might pick select molecules to test certain methodologies to construct bonds. As I mentioned, as a synthetic chemist, we can create wholly new methods to form specific bonds. Here's a particular example, actually developed by my first ever postdoc, Marco Weimer, who's in the audience somewhere. And you see, we developed this method to close this particular ring system to form this helical molecule, which will appear later in my talk. But the final reason we might make molecules is to use them for something. And by far and away, that's the largest reason that I'm interested in making molecules. I want to use them for something. Now, does that make me an applied scientist? Well, there's no doubt that chemistry is incredibly useful. Our ability to invent and create new molecules has transformed the modern world through the discovery of new drugs and diagnostics, to synthetic dyes, to agrochemicals, to components of electronic displays, and a wide variety of other applications. Chemistry is incredibly useful. But how important is it to be useful, to be important in science? Indeed, relatively early in my scientific career, there were some significant protests between scientists and one of the main funders of physical sciences in the UK, the EPSRC. Now, while there were many aspects of these debates and protests, I guess one of the major concerns from scientists was that the EPSRC was asking scientists to focus on short-term research projects with immediate economic impact. In other words, be very applied in their research, and scientists were concerned that this would deplete basic science research in the UK. Now, while I certainly understood many of the discussions that went on at this time, I was a little at odds with the debate, Because personally, I didn't see anything wrong with framing your research projects with a particular use in mind. But on the flip side, I didn't think that that negated the ability to do basic science research. In other words, I felt there was a middle ground between these two extremes of basic science and applied science. And indeed, other people have thought about this, arguably in far more detail than I have. And here's a very nice example from Donald Stokes, who proposed a quadrant-type model. You see, in this model, on one axis, we have the quest for understanding, basic science research. On the other axis, we have considerations for use, applied science research. And what's nice about this quadrant approach is, for those who do pure basic research, they're encompassed in this quadrant. For those who do pure applied research, are encompassed in this quadrant. But it's this particular quadrant that was most interesting to me. Research that's inspired by a particular use, but also requires significant basic science research. And this is a quadrant that I hope most of the research that's ongoing in my group is aimed towards. Indeed, what we aim to do is develop multidisciplinary projects whose impacts may well extend beyond the traditional boundaries of chemistry, but in turn, that tells us a lot about what we do and don't know about our molecules, and so inspires further fundamental study. And what I'm hoping to do in my lecture tonight is give two different case studies of multidisciplinary topics that my group focus upon, trying to exemplify this fact on being led by certain uses, but that in turn inspiring fundamental study. Before I do that, however, I thought I should give a quick summary of my research career to date, particularly for the younger members of the audience, to show the journey that I took and some of the decisions that I made. I thought I paused on that one so you could appreciate the picture. So I actually grew up not far from here in northwest London. My sister and I were actually the first in our family lucky enough to go to university, where I ultimately studied chemistry. But I think it's fair to say that when I was growing up, I wasn't particularly passionate about chemistry. Arguably, I wasn't particularly passionate about science either. Actually, my biggest passion growing up was music and the performing arts. That's what I really loved to do. So much so that when I was 14 years old, it came to picking the options I would take at my GCSEs, and I was originally intending on taking a relatively science-light set of options. And it was actually one of the teachers in my school on a school open day that said to my parents that I had an aptitude in science and should be encouraged to do the more science-intensive courses. So that's what I did. Of those courses, certainly chemistry was my favorite, but I still don't think in those days I was really passionate about chemistry. Indeed, when it came to taking my A-levels, I absolutely hedged my bets. So I took 4 A-levels, chemistry and maths, theatre studies and music. And ultimately, if I'm being absolutely truthful, I selected chemistry to study at university because I felt the employment prospects of a graduate chemist were better than the employment prospects of someone who graduates performing arts. Nonetheless, that led me to Bristol, and I had an absolutely wonderful time at Bristol, and that's certainly what generated my love of chemistry. And I think there's a number of reasons for that. Firstly, I don't think it's uncommon in a complex subject like chemistry, that only once you develop a deeper understanding of the subject can you start to see some of the creativity and some of the beauty and truly start getting excited by the subject. The second reason I think Bristol was so important to me is chemistry is a practical subject. It's a laboratory-based science, and I had some excellent research experiences at Bristol in the lab. The first of those was a summer internship at the pharmaceutical company GSK, where I worked on some mechanistic organic chemistry. And the second one was a final year research project with Chris Willis at Bristol, where I worked on an organic synthesis project towards the synthesis of a naturally isolated compound. And I absolutely loved my final year research project. So much so, I sought advice from Chris and from others on potential groups where I could do a PhD to work on natural product synthesis. That's what I wanted to do. With that in mind, I was delighted to be offered a PhD with Tony Barrett here at Imperial. Tony is very, very well known for his research in natural product chemistry. But it's there I made another slightly unexpected choice. When I started with Tony, he actually offered me two potential projects. One of those was a natural product synthesis, but ultimately I decided not to do that project and take a different project entirely, a slightly more niche area of Tony's research, these conjugated porphyrin molecules. And I think the reason I selected this project is I was really excited by the breadth of science that I could do within this project. There were certainly challenges in how we would build these molecules, how we would selectively synthesize them, but there was fundamental physical study of their photophysics and particularly their ability to sensitize oxygen. And finally, we were developing methodologies where we could attach these macrocycles to some kind of biological targeting vector, such as an antibody. So if I summarize some of the areas of work for my PhD and the topics I worked in, you see it spanned synthesis, optical and electronic properties, and biological chemistry. And it's amazing to see how my current research portfolio maps beautifully onto those three areas. How much the science that we do in our training further influences us, even subconsciously, in our independent career. Beyond my PhD, I went on to work with Andrew Holmes. There again, I worked on something slightly different, some synthetic methodology, developing reactions in a fluid-like state of carbon dioxide called supercritical CO2. This requires reactors to be pressurized, so I had to work with pressurized batch and continuous flow reactors. So there's quite a bit of engineering in addition to chemistry. But again, Andrew's group was very multidisciplinary. And so in addition to the project that I was working on, there was people in Andrew's group working on synthesis. Andrew's very well known for his work on organic electronic materials. And at the time, there were some really nice projects going on surrounding phosphoinositile chemical biology. So again, these three topics of synthesis, materials, and biology were really embedded into me, and that's what seeded my independent career. Now, what I'm going to do in the remainder of my talk is talk about two different multidisciplinary areas that are the focus of my group's research. The first one of those is our desire to use chemistry to interrogate biology. And this has been an area of my research that's been fundamental to my whole period at Imperial College, particularly in the area of therapeutics and drug discovery. Now, for those in the audience unfamiliar with how drug discovery works, I thought I'd talk you briefly through the logistics of so-called target-based drug discovery. What happens in target-based drug discovery is if we have a particular, see if I can get my pointer to work. I'll come over this side, shall I? No, I've got to stand here. Okay, you can see the unhappy face, right? If I identify a disease that I want to treat with a drug, what I can do is I can undertake biological research on that disease, trying to understand the biology of the disease. And through understanding the biology of the disease, hopefully we can identify a biological molecule, often something like a protein, that plays a key role in the biology of that disease. That then defines a target which we can aim to engage with our drug. We then go about finding molecules that can bind to that target and, importantly, disable that target. The molecules we initially identify then have to be significantly optimized, so extensive medicinal chemistry has to be done to optimize the strength by which this molecule binds to its target, how selective it is over all the other molecules in your body, its metabolism, its distribution, and so on. And if we do a good job, that will lead us to a candidate experimental medicine that can enter clinical trials. Then ultimately, if our hypothesis was correct, that the target was indeed critically important for that disease, ultimately our drug should improve the outcome of that disease. Now, this is a very logical approach to take, but it's not without its challenges, and it's not without its failures. And one of the main reasons that target-based drug discovery can fail is the fact that biology is complicated and it's busy. What this means is, if we're busy studying the busy biology of our disease, we might identify a suspicious-looking target. We think they're really important for that disease. We then can go about developing chemical tools to disable that target. But ultimately, through further study, it might transpire that the target we picked wasn't the right target at all, and instead we should have been looking for something else entirely. Nonetheless, what I would argue is the process of developing a chemical tool is incredibly useful to prove the biology of our selected target, its importance in that disease, and validate the approach. And so that's been a large part of my research here at Imperial College, trying to develop chemical tools to validate new disease targets. And the particular biological area of interest to me has been the process of transcription. So hopefully everyone in this audience knows what DNA is. This is our genetic blueprint of life, the biomolecule that contains all the instructions to make the chief biological molecules in our cells proteins via an intermediate state of RNA. Now, right at the start of the process where the information in DNA is decoded is transcription. These events act upon our genome, determine how, when, and in what context different genes are switched on or off. As such, these events are incredibly important in cellular identity, incredibly important in how cells respond to the environment and to stresses, and also incredibly important in the development and maintenance of human disease. Now, just like I said, biology is complicated, and transcription is no different. Here's a diagram from a recent review summarizing some of the events involved in regulating the transcription of this particular gene. And I don't expect you to take in much of this information. I just want to show the complexity of this process, and through that show there's a rich source of therapeutic targets. So part of my work at Imperial has been trying to validate some of those transcriptional therapeutic targets. And I think I've got a few case studies I can share with you that have been quite successful. So the first of which, Alan actually mentioned at the start, was a large-scale effort, together with Simak Ali and Charles Coombs and Tony Barrett, to develop the first inhibitors of an enzyme that regulates transcription, CDK7. We developed some of the first selective inhibitors of this target, and then subsequently optimized those into clinical candidate medicines that we licensed to a company who took them into clinical trials, and those clinical trials for cancer are ongoing. More recently, I've been working extensively with Hugh Brady, who's in the audience, and Simak Ali, now trying to drug a particular transcription factor. This is a molecule that directly binds to DNA and regulates transcription. We found that this transcription factor, RevERB, plays an incredibly important role in the development of certain immune cells. And so by inhibiting this transcription factor, we have a useful therapeutic approach in the area of immunotherapy. Cancer immunotherapy is a really hot topic right now, so we're currently trying to further develop this project through spinning out a company. Finally, I've had a long-standing interest in how DNA is packaged and targets associated with that packaging, particularly targets known as the histone lysine methyltransferases. So together with Bob Brown here at Imperial and Arthur Scherf at the Pasteur Institute, I've been involved in a number of different projects aiming to develop chemical tools of these particular transcriptional targets and using them to validate new approaches in diseases such as cancer and malaria. So these approaches, I think, have yielded useful molecules that can help validate these transcriptional targets in certain diseases. But I think the methodology we've used has been relatively conventional in these projects. We've used conventional organic synthesis and medicinal chemistry. So how can we use our fundamental understanding of chemistry to create even more precise tools to interrogate biology? Well, one particular approach that has been of interest to my group recently is trying to determine in ways and develop ways in which effectively we can control the ability of our small molecule. It doesn't like my pointer there. I can do it over here. Trying to control the ability of my small molecule, here it is, to bind to my target. So can I develop ways in real time to control how that binds to my target and therefore monitor that event relative to the downstream events? In other words, what we would like to do is develop a method that under certain stimuli, we can turn our drug on or off. And the particular stimulus that we're interested in is light. So if we can incorporate a light-sensitive component into our compound, we can use different colors of light to switch our drug into different shapes. If one of those shapes binds, we can switch our drug on here. If the other shape doesn't bind, our drug will be off. So we can use our different colors of light to switch our drug on or off. So what we need is that molecular component that changes shape when we use light. So we use one color of light, we change it to one shape. We use another color of light, we switch it back again. If we then encompass that in a larger system, such as a drug, what you see is that shape change drives a large shape change in our whole molecule, which in turn will relate to that difference in binding. Now, the molecules that people tend to try and use for this approach are the azobenzenes. These are molecules that have a nitrogen-nitrogen double bond and two aromatic rings, and they very nicely absorb light to switch between these two isomeric forms, the so-called E isomer and the Z isomer, dramatically changing shape in the process. Now, the azobenzenes work quite well, but they have some performance issues. So this spurred some further fundamental study in my research group to try and improve the azo photoswitches. And ultimately, we did an in-depth study of replacing one of those benzene rings for a 5-membered heteroaromatic ring system. I think we learned a lot along the way in this study, but I think the most useful molecules we discovered were the so-called azopyrazoles, where one of my benzene rings has been replaced by a pyrazole ring. In doing that, we get dramatically improved photoswitching, and we get improved stability of our switchable states. So in other words, we developed a vastly improved molecular component that we now want to employ in a switchable drug. So I'm going to show you one particular example of doing this. And this was a project together with Randy Peterson's group at Utah, where we were aiming to develop photoswitchable inhibitors of a particular ion channel called TRPA1. This is an ion channel that's present in cell membranes. It's actually responsible for sensing irritants. And together with Randy's group, we developed a photoswitchable ligand for this ion channel. Now, these ion channels are partly responsible for membrane potential in cells. And so the data you see on the bottom right-hand side here, this is electrophysiology data where we can measure current voltage characteristics in cells upon treatment with our compounds and now exposure to light. So these are cells that express our target and have been treated with our small molecule. And what you see is this violet line is a flash of violet light. That switches our compound on, which gives a dramatic change in the current. That is then sustained, and you see a flash of green light. That then switches our compound off, and the current returns to the normal state. So we can use light to switch our drug on and off. That then activates or deactivates the channel and has an effect on the current that we observe. So that's a cellular study. How can we show that in an even more dramatic outcome? Well, Randy's group have developed a very nice zebrafish model where they have engineered zebrafish that express our target of interest in the cardiomyocytes, so the muscle cells of the heart. We can then treat these zebrafish with our photoswitchable compound, and then we can observe how those zebrafish respond to different colors of light. And so what you're going to see now is a movie of live zebrafish embryos that have been treated, zebrafish larvae that have been treated with our compound. So at the moment when we treat these embryos, the compound is in its off state. So those hearts beat away as you might expect. Now what you're going to see is a flash of violet light. You won't be able to tell the color, but it will be violet light, and that switches the compound on. The pharmacological action of the compound effectively stops the heart beating, particularly in the ventricle component. On the right-hand side, you'll see a second flash. This is now green light. That switches the molecule off again, and the heart starts beating again. So by incorporating this photoswitchable component into a ligand for a biological target, we can control whole animal physiology using light. And this is tremendously exciting for all kinds of studies, for excitable cells in neurology, for studying kinetic processes in biology, and potentially for new approaches in therapies. The second example I wanted to discuss was my interest in using chemistry to invent new materials. And in this area, I guess my focused area of interest is in the property of chirality. This is a property of symmetry or shape of objects, tiny through to large objects. It was first defined by Lord Kelvin, who said, I call any geometrical figure or any group of points chiral and say it has chirality if its image in a plane mirror, ideally realized, cannot be brought to coincide with itself. In other words, chiral objects exist as a pair of non-superimposable mirror images. And so an excellent example of chiral objects that hopefully you can all appreciate are your hands. If we examine our hands, they are mirror images of one another. If you imagine a mirror plane down the middle, they're mirror images of one another, but there's no way you can superimpose your hands upon one another. They are non-superimposable mirror images, so they are chiral objects. And they exist in a left and a right-handed mirror image form that we would call enantiomers in chemistry. Now that's all well and good, but how useful is chirality? How can we exploit this property of chirality? Because it's relatively subtle. Indeed, if I stick to the example of my hands, and I consider the interaction of my chiral hands with a non-chiral object, such as this glass, the interaction with my left hand and what I achieve with that interaction is the same as what I can achieve with my right hand. A chiral object interacting with a non-chiral object is equivalent. But chiral objects are sensitive to other chiral objects. And to demonstrate this, I'm going to need a volunteer, so I'm actually going to ask Professor Armstrong just to come up again briefly. I didn't actually tell him I was going to do this. So now if I want to use my chiral right hand to shake Alan's right hand, we can shake hands, as you might expect. But now if I use my chiral right hand to shake Alan's left hand, it becomes a little bit awkward. In other words, the process of shaking hands is sensitive to chirality. We only get a productive interaction if we both use our right hands. Thanks, Alan. Can we give a round of applause? So this sensitivity of one chiral object for another is used in certain applications, particularly in the areas of biologically active molecules. Because life itself is chiral, the molecules that make up our biomolecules in our body are made from building blocks that are chiral, and moreover, they are so-called single-handed, so they only exist as one of those mirror image forms. For example, amino acids are all left-handed. What this means is, when we go about developing a molecule that will interact with a biological system, those molecules in that system will be sensitive to the chirality. So here's a very nice example of a molecule called carvone. You see the structure of this molecule on the right-hand side. The only difference between these two structures is that they are mirror images of one another that are non-superimposable. Nevertheless, if you were to isolate these two mirror images and smell this one, it would smell like spearmint. But if you smell the other one, this one here, it would smell like caraway seeds. The only difference is they are mirror images. Otherwise, they're identical. And the reason they have such different smells is because the receptors in your nose are chiral, and so they are sensitive to the chirality of the molecule that binds to those receptors. So this approach in biologically focused molecules is exploited in drug discovery, agrochemicals, and a number of different applications. But I've become interested in, would chirality be useful for other areas, particularly areas of technological importance? And this has become quite a big topic for my group. And I'm going to explain the approach to you, but first I need to talk about one other aspect of chirality, no longer chirality of matter, but now chirality of light. So hopefully everyone in the audience, or at least a fair number of you in the audience, have heard of linearly polarized light. This is light polarized in such a way that the wave is confined to a single plane. Linearly polarized light is exploited in a number of different applications. Perhaps one most familiar to you will be in polarized sunglasses. These exploit the physics of linearly polarized light to reduce glare. But I'm interested in an alternative polarization state of light, so-called circularly polarized light. This is now light that's polarized such that the wave traverses as a helix. And helices are objects that you observe in the natural world all the time: screws, springs, shells, pasta, all kinds of examples. And because of the twist of the helix, they exist as mirror images of one another, but they are non-superimposable upon one another. So the helix, as an object, is a chiral object that exists in a left and a right-handed mirror image form. Now, because circularly polarized light is helical light, it's effectively chiral light, and also exists in a left and a right-handed mirror image form. Now, actually, circularly polarized light crops up often in the natural world also, and I'm going to give you a key example of that. One of the examples emerges from animals that have beautiful iridescent colors. And there's a number of different animals. Here's an example of beetles, but there's a number of different animals, peacocks and so on, that have iridescent colors where those colors don't emerge from pigments, they emerge from structural effects on the surface of that animal's body, so-called structural color. These structures give rise to interesting optical effects that give rise to the iridescent color. Now, taking these beetles as an example, they actually use a biopolymer, and they arrange chains of this biopolymer. They arrange them on top of each other, forming a helix. Now, the helix that they use selectively reflects light, and via the physical phenomenon of Bragg reflection, gives rise to structural color, the beautiful iridescence you see. But remember I said a helix is a chiral object. And so because this beetle is using a chiral object to reflect the light, turns out the light that's reflected is chiral. Particularly, it's left-handed. So this beetle selectively reflects left-handed circularly polarized light. So I'm going to show you a demonstration using a very expensive bit of kit, some 3D cinema glasses. Those of you who have been to the 3D cinema, modern-day 3D cinema actually uses circularly polarized light to differentiate the image that each eye receives. And so 3D cinema glasses are really nice, cheap, circularly polarizing filters. So here's one of those beetles. Remember I said that these beetles selectively reflect circularly polarized light, left-handed light. So I can use my glasses, and I can look through the left circularly polarizing filter. Let's see if I can do this. And if I do that, what you should be able to see is the beetle remains green. It's a pretty unexciting experiment. Alternatively, though, if I now switch to use the right circularly polarizing filter, the beetle turns black. So we use one, it's green, one, it's black. And that's because in one of those circularly polarizing filters, the left-handed circularly polarized light cannot travel through the filter, so we lose that structural color and the beetle becomes black. Now there's an interesting feature of the physics of circularly polarized light, and that's the fact that when it reflects off a reflective surface, it changes its handedness. It inverts sign. So left-handed light becomes right-handed light. And so we can also show this using a mirror. So now we have our beetle. Remember, the beetle itself should be reflecting left-handed light, but the light that reflects off the beetle and then off the mirror should now be right-handed. So if I use my left polarizing filter for this, what you should see is the beetle in the mirror is black, or at least pretty dark, and the one on the other side is green. If we switch it over, we get the opposite effect. So I could keep doing this all evening. And that physics of circularly polarized light is incredibly important for one of the technological applications I'm going to talk about in my talk. Before I move on to that, I should say that I believe there's still some debate as to whether beetles actually use the fact that they reflect circularly polarized light for any behavioral activity. But there are certainly some animals that appear to be sensitive to circularly polarized light and use it in their behavior. So here's a nice example of a shrimp. This particular shrimp's eyes are sensitive to circularly polarized light, so these shrimps can use circularly polarized light to send secret signals to one another without attracting the attention of predators. So I've been mentioning a lot about technology, and the technology that I'm going to focus on are organic light-emitting diodes, OLEDs. These are little devices where an organic material, go over here, are we? This pointer's got a mind of its own. An organic material is sandwiched between two electrodes. One of those is transparent. If we apply a potential across those electrodes, our organic material glows. It generates electroluminescence. That's why an OLED lights up. And these OLED devices have become incredibly useful for a variety of applications, but most notably in displays. So many state-of-the-art smartphones, for example, now use OLED displays. And OLED displays are really, really good in many senses, but historically they have one problem, and that's the fact that there's many reflective components in those OLEDs. So if you take your phone outside on a bright sunny day, the external sunlight would reflect off those reflective components, and you wouldn't be able to see your screen. You get a significant amount of glare. Now, the way OLED manufacturers have got around this process is generating anti-glare filters. And interestingly, the anti-glare filters they use exploit the physics of circularly polarized light. So what happens is, you take your phone, for example, out on a bright sunny day. The external light travels through the anti-glare filter, which converts it into circularly polarized light of a certain handedness, in this case, right-handed light. If that circularly polarized light reflects off a reflective surface, it changes sign. Right-handed becomes left-handed light. And now that light's of the wrong sign to pass back through the anti-glare filter. So none of the light that enters your display can get out again. And that's why this works as an anti-glare filter. So this is a beautiful solution to glare, but it has a significant problem that within your display you have your OLED pixels. They generate non-polarized light, and only 50% of that light can get out past the anti-glare filter. So what OLED manufacturers have done is they've taken a 50% cut in energy efficiency to make sure your screen is more viewable in bright light conditions. Now, in theory, you could tackle this problem if you could generate OLEDs that directly emit circularly polarized light. Now that light should be able to get through the anti-glare filter without any loss, and you would have a more energy-efficient OLED display. However, when we started our work in this area, and this was in collaboration with Alistair Campbell in physics, it wasn't clear how one could generate organic materials that would emit a significant amount of circularly polarized light. And we discovered one approach that I think is pretty useful. And this approach uses conjugated polymers that are already established for use in OLEDs and chiral small molecules. And I'll talk you through the approach. So first and foremost, this approach, we used a conventional conjugated polymer, this polymer here termed F8BT. If you make thin films of this polymer, you get this nice bright green emission from those films. But this polymer is non-chiral. So you wouldn't expect the light emitted from the polymer to be chiral. And indeed, this plot on the right-hand side is a measure of how circularly polarized the light that's emitted is. As you can see, it's pretty much a flat line. A non-chiral molecule gives non-chiral light. But what we found is if we blend in a small amount of a single-handed molecule, in this case a right-handed molecule, to those thin films, what we get is a material that still emits green light, but now that light is significantly circularly polarized, and in this case it's significantly right-handed. So what's happened here is the chiral small molecule has induced chirality in the polymer that's then resulted in chiral emission from that polymer. And we know the small molecule is responsible for the effect because if we use the opposite mirror image, the opposite enantiomer of that small molecule, we see the opposite effect on the light. Now a left-handed molecule gives rise to left-handed emission from the polymer. So this in principle is a very simple way to turn a conventional OLED material into a circularly polarized emitting material. And so we've been working extensively over the last few years trying to improve the effectiveness of this approach, trying to give state-of-the-art devices that could realize this approach, hopefully one day in displays. But along the way, this use-inspired research has told us what we don't understand about the system. And here's a particular piece of data I want to share with you. This is data from one of our recent papers comparing electroluminescence and photoluminescence from our materials. You can focus on one of those, it doesn't matter which, you see a similar effect. I'm going to walk you through this data. Let's focus on the red line. The red line is from our material where we have our polymer blended with the right-handed small molecule. When our films were about 100 nanometers thick, the light is significantly right-handed, but when we double the thickness of our film, the light becomes significantly left-handed. What we've done is change the thickness of the film. The chirality of the small molecule is identical, but the chirality of the light is completely inverted. This is fascinating, and we don't fully understand it. So this has inspired a lot of current fundamental study in my group to try and elucidate the factors at play. In turn, this area of research has sparked a number of different application areas for my group interested in this use of molecular chirality in technologies, from devices that can emit circularly polarized light to detect circularly polarized light to tune bulk properties using chirality. And anyone interested in this area, you can take a look at this relatively recent review that I wrote together with Joachim Brand and Francisco Salerno. Here we cover some of the potentials of chirality in technological applications, from polarized light to bulk effects, and increasingly how one might be able to use chiral materials to control the spin of electrons. Well, with that, I think my time's just about up, so I want to conclude from my talk. What I've hoped to do in my talk is convince you that as chemists, because of our understanding of molecules, molecular structure and properties, we have unique insight that we can employ to study a number of different areas. And I would argue that particularly our ability to invent and make wholly new molecules is incredibly enabling to pursue these areas. And hopefully I've given you some nice examples, from new drugs to treat diseases, to ways in which we can harness the power of light in biology, through to chirality in materials science. I'd like to conclude by thanking a few people. First and foremost, I'd like to thank my research group for all their hard work. As I mentioned, this is a laboratory-based science, and these are the people that worked hard in the lab to realize all these research outputs. I'm immensely grateful for all my group, for all the work that they've done. Many of them are in the audience tonight. I'm incredibly proud of what they achieved in the group and what many of them go on to achieve since they've left the group. When doing multidisciplinary science, collaborators are key, and almost without exception, every project that my group works on is in collaboration with other scientists from other disciplines. And here's a selection of my most long-standing collaborators. I should say, if you're in the audience, if you're a collaborator and you're not on this screen, I'm going to apologize now. I appreciate you also. But I really want to say thank you to all my collaborators. Incidentally, I've noticed that I tend to collaborate with people whose surnames are at the start of the alphabet. So if your surname is at the latter end of the alphabet and you fancy a new project, I think come and have a chat, we can balance this out. I couldn't do the research I do without significant research funding. I'd like to thank all these people for funding the research that's ongoing in my group, particularly the EPSRC and the Leverhulme Trust for funding my chiral materials research, and some of the cancer charities such as CRUK and Ovarian Cancer Action for funding my medicinal chemistry work. And finally, I'd like to thank the most important people in my life, the people who make everything worthwhile. So with that, I'll conclude, and I'll thank you for your attention.
Thank you, Matt, for an amazingly broad lecture ranging from photophysics to biology of zebrafish and beetles, with chemistry right in the middle. Matt has kindly agreed to answer questions from the audience. So if anyone would like to ask a question, please put up your hand, and we have, I think, some microphones somewhere that can be brought to the questioners. It's very hard for me to see down here who's putting their hand up because of the bright lights, but I'll try. We have a question over there. Get a microphone over to the far side. Just over there.
That was really... Is that on? Can you hear me?
I can hear you. I can repeat your question
okay.
If that's helpful.
So that was a really interesting talk, so thank you very much. You covered a really great amount of work. I wanted to ask a slightly technical question. So with your helicene molecules, so obviously those are chiral, and I guess you're having to produce those on quite large scales. So I was wondering, how do you actually get the single-handed enantiomers of the molecule that you're putting into these OLED displays?
No, it's a very important question. Something that I've glossed over in my talk that I may or may not get stick from some of my organic colleagues for is I haven't talked much about the synthesis that underpins these projects. And I should say for that particular project, one of the reasons we use that particular helical molecule is because we developed a very effective way to synthesize it on scale. Or I should say Marco developed that way. So we could make gram scale quantities of it, but as you've obviously recognized, that's as a mixture of the left-handed and right-handed form. What we tend to do is just separate those using chiral chromatography. Ultimately, for this application, people often say to me, oh, well, why don't you try and synthesize one over the other? In synthetic chemistry, that's a great challenge. But because we often want both handedness of the light, actually we want both enantiomers, so separation is a great option. What we ultimately would like to do is more scalable ways to do that, so selective recrystallization or so on, to separate these things, and I think those could be developed if one wanted to manufacture these. But ultimately, separation is the approach we take, yeah.
Any more other questions?
Can I ask one?
Andrew. Yes, microphone will be coming.
Matt, the very interesting talk, and the question of the deposition of your circularly polarized materials and the switch from showing one-handedness to the other is, I think, Bert Meyer's group originally showed polarized emission, and it always puzzled me because the challenge is you're going from a solution, I assume, to the solid, and it's in that actual morphology of change from solution to solid that feature happens. And you've shown a really interesting example, but I've always wondered how can one be sure to control it and make it reproducible?
No, it's a great question. And absolutely, in some of Bert's research, they've showed the importance of these kinetically formed structures. So ones that emerge out of that deposition process, but aren't necessarily in a thermodynamic endpoint, but they've been trapped. And it's that trap state that's incredibly important. Now, I should say that something that people often ask about those blend materials is the nature of the interaction between the helicene and the polymer. At least in even relatively concentrated solution, we see no evidence of a strong interaction at all. So you're exactly right. This process of induction of chirality happens as we're depositing the film and as these are becoming more concentrating and depositing themselves into films. Now, the approach we've taken is to do a lot of studies varying the blend ratio and the approach we take, annealing time, for example, annealing temperature, to develop a protocol that can robustly give us the maximal effect. Looking at Jess, because Jess Wade in the audience did much of this work, to develop robustly films that can do that. What we don't yet understand in depth is the structures that are formed and why they give such large circularly polarized light. And so that's the focus of a lot of our intensive spectroscopic work currently, is to figure out what we're forming in those. But the procedure is quite robust. We just don't fully understand the nature of the structure that's formed.
So a question over here. You can get a microphone over to this side, please. Thank you.
Thanks. It was inspiring and really brilliant. I'm not going to ask a question about anything I actually know anything about, so here goes. When light transmits through a solid, transparent solid, its speed slows because of a variety of interactions between the light and the solid. When circularly polarized light transmits through a solid, does it maintain its handedness?
I think the simple answer would depends on the solid. Honestly, it would depend on the solid. So, for example, it would depend on whether that was a solid made up from a chiral material or not, or whether, even if it wasn't a chiral material, if it had solidified in a chiral structure, so some non-chiral materials crystallizing chiral space groups that can be separated. And so those chiral solids would certainly have an effect on the circularly polarized light. In other cases, you would see an analogous effect to what you see with unpolarized light.
Yeah. Thanks.
Yeah, one more question just behind. Just behind that.
Hello. When you have the filter where the light goes in and reflects the other polarization, so cannot escape, have you got any problems with overheating of the base material if you're trapping light in?
It's a good question. Maybe that's why they write on you shouldn't leave your phone out in bright sunlight. I honestly don't know as to what the thresholds would be. I suppose, yes, you would have a heating effect. But I don't know to what extent that causes a problem. But yeah, I think your thinking is right as far as I know. But I don't know to what extent that changes the usability of the screen.
So I think in view of the time, we'll bring the Q&A to an end. Matt will be, I'm sure, happy to answer questions later in the wine reception. But to close the session off, I'm delighted to invite Andrew Holmes to provide the vote of thanks.
Well, as mentioned by Alan, my name's Andrew Holmes, and I had the real privilege of being a co-worker with Matt when he was in Australia. There's a bit of a backstory to that. I won't go into the details, but Matt ended up coming to Australia because a postdoc that had been lined up didn't materialize, and through Tony Barrett, CSIRO, who are the government national labs you heard about, were able to offer Matt a position, and I was one of the lucky beneficiaries of having a collaboration with CSIRO where they provided some postdocs to collaborate with. And what a wonderful experience it was to have Matt arrive, I think in October 2006. And we'll get him back, and we're getting him back, you'll see, you've already heard. But by June 2007, he was back here. Now, he was remarkable. I have never, ever seen anyone in a research group who understood everything that everyone was doing in the group and asked a penetrating question and then provided a brilliant original solution. So that was a fantastic experience, just to watch this performance, not only intellectually, but in the lab as well. In that period, from October 2006 to June 2007, you were the co-author of 3 papers. I think that's pretty fantastic as well. And it was wonderful to be a co-author with him. And you've heard the kinds of research interests that he has developed. And he's speaking to the converted, of course, so I declare a conflict of interest. But having that spectrum of interests from medicinal chemistry, and in Australia he worked in medicinal chemistry, synthesis and supercritical CO2, and he saw all the problems about organic electronic materials. And that was just quite an experience to be able to enjoy. Coming on now to some topics in tonight's lecture. Again, I think Mac raised a really interesting question which all of us wrestle with. The issue of curiosity-driven science, or science that's target-oriented in some way. And there's no perfect solution. It's something I've been thinking about a lot, and I'm sure everyone else in the audience has too. And I'm going to have to give a talk in the Australian Academy of Science next May on the topic of the role of chemistry in the issue of doing science for the public good. And if any of you have any great ideas, I really appreciate them. Struggling, because it's very easy after the fact to go back and say, well, you know, lasers were invented. Who would have thought they were going to be useful? And not that that's specifically chemical, but just to take a typical example, things that were discovered by curiosity-driven science that turned out to be hugely interesting. But what we can't, most of us can't do, I think, is predict what's going to be hugely interesting. And perhaps that's the reason we do it, because we like having discovery by surprise. So I thought that was a really interesting introduction, Matt, and thank you for that. And then this challenge of doing target material for cancer research and for transcription and so on, and then organic electronic materials and circularly polarized light, and how you get something out of that that's really meaningful. So I want to congratulate you also, because I felt you certainly made it possible to understand what you were doing. Don't you agree? It really was something we could all follow. So thank you for that. That's a great talent and will be very valuable as you go on. Congratulations on your appointment as a professor. Now you have to profess. And I think one of the thrills of being an academic is actually to work with someone who's much better than you, and then to be able to bask in the glory of their successes. And that is fantastic. Family all must be hugely proud of your achievements, and I'm sure, as you showed in the last slide, it's a two-way process. So, ladies and gentlemen, I'd like to propose an official vote of thanks to Professor Matt Fuchter. Would you please join me in congratulating him?